Hello everyone, thank you for joining me today. My name is Tyler Kelleberger, and I am excited to tell you a little bit about a gene known as MRDC1, which works to inhibit dendritic cell maturation. For a brief background of the study, glioblastoma is the most common and lethal malignant brain tumor in adults. One potential and experimental treatment approach is immunotherapy, or using the body's natural immune system defenses to fight off cancer. Dendritic cells, or DCs, are a critical immune cell subset linking the innate and adaptive immune responses. Though well characterized, there is still a need to better understand basic regulatory mechanisms of DCs, which may lead to better immunotherapies in the future. Researchers in our lab used a deep learning-based platform to identify 9 master regulators of dendritic cells shown in the table below. One such factor we discovered was MRDC1, a factor never described in the context of dendritic cells. Preliminary analysis showed high expression of MRDC1, specifically in dendritic cells, which is shown in the graph to the right of Table 1. We hypothesized, therefore, that MRDC1 regulates DC maturation. For the methods of the study, we first characterized DCs in a murine model using wild-type and MRDC1 knockout mice. Next, we measured levels of co-stimulatory molecules and MHC1 in knockout DCs compared to wild-type DCs. We also completed a restimulation assay to assess DC function, and then completed RNA sequencing using bone marrow-derived DCs to identify important gene networks. Lastly, we investigated tumor growth in a GL261 brain cancer vaccination model with luciferin luciferase. For the results section, starting with DC characterization in our murine model, we extracted splenic DCs from knockout and wild-type mice, and observed no difference in DC proportion in mice at homeostatic state, as shown by the first plot. In the second plot, you can see that knockout DCs expressed higher levels of co-stimulatory molecules, as indicated by CD86 and CD80. In the third plot, increased MHC1 expression was observed in knockout DCs compared to wild-type DCs. Moving on to the restimulation assay, we first generated wild-type or MRDC1 knockout bone marrow-derived dendritic cells for 7 days, electroporated OVA RNA into DCs on day 8 so they can present it in MHC1 or MHC2. Then we injected these cells into wild-type Black 6 mice. After a week, we harvested splenocytes, T cells and other splenocytes, but only the T cells would secrete interferon gamma, and co-culture them with melanoma target cells secreting OVA, B16F10 OVA, which is a melanoma cancer cell line that secretes OVA. We then analyzed the interferon gamma response using the ELISA technique and looked at T cells with flow cytometry. These results showed greater interferon gamma secretion with knockout DCs into wild-type mice than with wild-type DCs into wild-type mice, shown in the ELISA graph of this section. And this means that knockout DCs activate T cells more. We also observed an increased activation of both CD8 and CD4 T cells with knockout DCs in comparison to wild-type, as shown in graphs 2 and 3 of this section, respectively. After the restimulation assay, our lab decided to conduct RNA sequencing using bone marrow-derived DCs, and we did this to identify important gene networks that may be involved. The RNA-seq results identified significant increases in knockout DCs in gene networks that have been shown to regulate DC maturation, such as CD83 and CCR7, shown below in graphs 1 and 2 of this section. The RNA-seq also identified increased expression in wild-type DCs of a gene which encodes an immune inhibitory receptor ligand, CD274, or PDL1, which is shown in graph 3 of this section. Lastly, we completed a brain cancer vaccination experiment with GL261 cells, using luciferin luciferase enzyme to visualize tumor growth. The glioblastoma cancer cell with luciferase enzyme was first injected into either wild-type or knockout mice. Both knockout and wild-type mice were divided into 2 groups, either no-flank vaccination group, which received only the intracranial IC injection, and vaccination group, which were injected in the flank first, then IC after a week. We then observed luminescent signaling in vivo. The results from this experiment demonstrated that prior flank vaccination against GL261 led to a significant reduction in orthotopic tumor growth in knockout mice compared to wild type, shown by decreased luciferase signal or less bluish color in the photos and plot below in knockout mice. In addition, prior flank injection against GL261 also led to a significant reduction in death in knockout mice compared to wild type, as shown in the survival plot in the top right. For conclusions, knockout DCs expressed higher levels of co-stimulatory molecules and MHC1 compared to wild-type DCs. Restimulation assay results demonstrated that knockout DCs lead to increased proportion of activated T cells that possibly secrete more interferon gamma. RNA sequencing identified significant increases in knockout DCs in gene networks that regulate DC maturation, as well as PDL1 in wild-type DCs. The GL261 brain cancer vaccination model demonstrated that prior vaccination against GL261 led to a significant reduction in orthotopic tumor growth and death in knockout mice compared to wild-type mice, indicating an augmented adaptive immune response in the absence of MRDC1. Altogether, these findings suggest that MRDC1 may act as a brake to excessive immune activation, and its inhibition may enhance DC function in therapeutic modalities in the future. Finally, I would like to acknowledge and thank everyone in the Tran laboratory who contributed to this project, and give a special shout out to Matthew Sebastian for his help and mentorship. I also want to thank the UFHealth Cancer Center University Scholars Program for their support in regards to funding and training. Thank you for your time and attention.